Hello and welcome to BioBiz Buzz, the podcast where we explore how cutting-edge biology is being translated into tomorrow's medicines. I'm Mike Ward, and today we're looking at one of the most critical but underappreciated contributors to disease in the eye, the blood-retina barrier. When these specialized endothelial interfaces fail, you see problems like vision loss in diabetic macular edema and age-related macular degeneration. Our focus is Neuvasq, a company building first-in-class biologics that aim not just to block downstream pathology, but to actually restore barrier integrity, and doing that by precisely activating βcatenin signaling through the GPR124 Reck co-receptor complex that's on retinal endothelial cells. Joining me are two guests, Dr. Laufer, Chief Scientific Officer at Neuvasq, who brings decades of CNS drug discovery experience from roles at Teva and Lysogene, and he now leads Neuvasq's multispecific antibody programs. And also we have Dr. Wai Wong, who's a leading retinal clinician and scientist, who again brings the frontline view of how current therapies are helping, and also, I guess, where they're still falling short. So in this episode, we're going to unpack the scientific and clinical challenge, what Neuvasq's approach has achieved so far, and how the company plans to move forward. So Ralph and Wai, thank you very much for joining me. So to kick off this conversation, let's first get an understanding of the clinical problem and the unmet need. So Wai, starting with the clinic, when you see patients with diabetic macular edema or wet AMD, besides the fact that these diseases reduce their vision, how much do you see those conditions as diseases of the blood-retinal barrier?
Hello, Mike. It's great to be here, and thanks for your question. In patients that I see with diabetic macular edema or wet AMD, the breakdown of the barrier is a key feature of disease pathology in both conditions. Healthy blood vessels are needed to convey blood containing oxygen and nutrients to tissues where they're needed, and this is done in a controlled manner. However, when the blood-retinal barrier breaks down in disease, contents from the blood like fluid, lipid, and blood itself can leak abnormally into the retina, causing it to become swollen, become exposed to lipid deposits and blood itself, which are harmful, and as a result, suffer deficits in this much-needed supply of oxygen and nutrients.
Right. So, talking about that blood-retinal breakdown, what does it actually look like in practice, in terms of leakage or edema or neovascularization, avascularization, and how do those clinical signs, I guess, translate into visual acuity loss and long-term outcomes, I guess, for those patients?
Yeah, that's a great question, Mike. These signs can be detected at the retinal doctor's office by retinal imaging studies, and these include fundus photography and OCT imaging. And leakage and edema can be seen as a swelling and a structural disruption in the layers of the retina. And we see also fluid accumulating in abnormal cysts that are not present in the healthy retina. And new vessels or neovascularization can also be observed as new blood vessels that invade above or into the retina itself, and is often seen with bleeding and lipid deposits. And so what this happens is that this really disrupts the ability of the retina to function. The retina works similarly as a photographic film when you load into the camera. It enables good vision when it's lying flat and sensitive to light. So when it becomes swollen and structurally disrupted, then it loses sensitivity, and patients experience vision that appears blurred, distorted, and dimmed, and instead of being clear and crisp, like what you would expect. In the longer term, these structural changes can lead to the death of retinal cells and also the scarring of the retina, which is permanent in nature. As a result, vision loss can be permanent in long term and cannot be reversed with treatment.
So we know that the anti-VEGF agents, they've been sort of transformative as they reduce the degradation of vision acuity. But clinicians still see non-responders, recurrent disease. So why specifically do those current therapies fail to restore patients' vision acuity from your experience?
Yeah, the way in which current anti-VEGF treatments work is that they bind and neutralize VEGF, which is a key factor that's abnormally elevated in patients with diabetic macular edema or wet AMD. And VEGF contributes to the barrier breakdown in its own way. However, current treatments do not inhibit all the factors that contribute to breakdown, nor do they decrease the continued abnormal production of VEGF. So as a result, some patients, while they do respond to VEGF to some extent often, they fail to respond fully to anti-VEGF treatments alone and may require treatments that include additional modes of action to fully as well as durably restore barrier function in order to improve vision. So these are the ways in which they might be falling short.
Right. Okay. And we're going to come back to, how Neuvasq is looking at sort of new approaches. But I'd like to actually sort of focus a little bit more on the blood-retina barrier. So, Ralph, for listeners who don't live and breathe the biology, can you briefly explain why, so for example, beta-catenin signaling is so central to retinal vascular development and barrier formation?
Thank you, Mike, and thanks for the opportunity to be here. So to answer your question, Wnt beta-catenin signaling is really the master regulatory pathway for formation of the blood-retina barrier and also of the blood-brain barrier. And it is beyond doubt the most important driver of endothelial barrier development in the retina. Wnt signaling does is to establish the molecular architecture required for highly selective vascular permeability by directing retinal endothelial cells to acquire barrier-specific properties. And these include assembly of tight junctions, suppression of transcytosis, and expression of specialized transporters. And both human and animal genetic evidence as well as preclinical data consistently show that when beta-catenin signaling is disrupted, then barrier formation fails and vascular integrity is profoundly compromised.
So, previous development work has shown that canonical Wnt beta-catenin is required for retinal angiogenesis, and that Norrin and Wnt7a7b are essential for forming proper BRB vessels and also in a barrier endothelium. How did those insights lead you to the idea that Wnt agonists could therapeutically reawaken those pathways, and therefore repair leaky barriers in adult retinal diseases?
Yeah, so as you say, given the fundamental and indispensable role of Wnt signaling in neurovascular barrier biology, it had been evident for decades that developing safe and efficient methods to activate this pathway in barrier endothelial cells would be transformative for repairing defective barriers in retinopathies as well as neurological disorders. Such an approach promises to restore the core molecular programs that govern tight junction integrity, vascular selectivity, and endothelial stability. The true turning point here came with the genetic identification and mechanistic elucidation of specialized Wnt receptor complexes, which really created the foundation for rational, precision-guided therapeutic designs with the required cellular and tissue specificity.
So, Wai, from your perspective, how has the growing understanding of BRB biology influenced the way that clinicians think about these diseases? Has it changed how you interpret treatment response or disease progression in practice, besides, for example, the visual acuity measures?
That's a great question, Mike. The growing understanding of how not one but multiple biological pathways can influence and regulate the blood-retinal barrier has been really exciting and has led clinicians to consider the roles of therapeutic targets beyond VEGF in driving an optimal response in patients. So in addition to just looking at visual acuity outcomes in the long term, this broader understanding has prompted physicians, including myself, to consider how differently mechanisms can contribute to restoring blood barrier breakdown and reducing retinal swelling in the way that's most efficient, that's happening most quickly, and most durably with minimal recurrence and minimal injection burden. We're understanding this optimal structural response and the avoidance of recurrent swelling in between injections is really important in avoiding retinal cell death and also avoiding retinal scarring, and really leads to maintaining visual gains in the long term. So in short, these mechanisms really open up our perspective to looking at treatments in a new way.
So, Ralph, I want to come back to the sort of foundation of the company. So, if we sort of look at particularly sort of the literature published by Neuvasq's founder, Benoit van Hollebeck, that basically it was that, Wnt signals, via a very specific receptor complex, GPR124 and Reck, and that you can engineer Wnt7a variants that are selective GPR124 Reck agonists, and that these can activate the beta-catenin pathway, safely normalizing blood-brain barrier function in glioblastoma stroke models. From this foundation, however, how did Neuvasq decide to focus on GPR124 in retinal diseases such as wet AMD and DME?
So the founding university laboratory, the lab of Benoit van Hollebeck, had indeed rigorously dissected this pathway for more than a decade, generating an exceptional depth of mechanistic understanding of how to selectively engage Wnt signaling in barrier endothelial cells. And this foundational body of work was published in Science and Nature papers, underscoring its disruptive scientific significance and broad recognition. By the time Neuvasq was launched, proof of concept had already been established that selective activation of this pathway was not only feasible but achievable with translational relevance. And GPR124 emerged as a preferred target from this work, given its fundamental role in Wnt signaling, which is one, as we said before, is one of the major drivers of neurovascular barrier formation and maintenance in the retina and in the central nervous system in general.
Yeah, okay. So can you describe in practical terms what a GPR124 agonist is actually doing at the level of the endothelial cells in the retina?
So our GPR124 bispecific molecules bind to both GPR124 and to another Wnt7 co-receptor called LRP6. And these engineered multispecific antibodies are conceptually elegant in that they closely mimic the activity of the endogenous Wnt7 ligand by engaging the receptor complex in a nature-inspired manner, like Wnt7 itself, that respects the pathway's intrinsic biological logic. And we hypothesized from the beginning that this design, so directly engaging GPR124 and LRP6, would maximize both potency and safety by preserving the physiological modes of receptor engagement rather than forcing artificial activation states. And in the end, its prediction was indeed borne out. The antibodies not only behaved as intended, but really exceeded expectations in terms of potency.
Right, yeah. I mean, as I understand it, that your lead candidate, NVQ401, is actually a highly potent GPR124 LRP6 targeting bispecific antibody. So I'm just wondering, how does this mechanism of action actually translate into patients' vision and acuity improvement?
This is an excellent question, and the answer to this is that we can build here directly on the extensive body of evidence that was generated by many laboratories worldwide, demonstrating the barrier-promoting functions of Wnt signaling. Across the blood-brain barrier and the blood-retina barrier, the Wnt beta-catenin pathway is a central determinant of endothelial specialization, because it drives the formation of tight junctions, which are important for the passage of small molecules across endothelial cells, so from the blood into the retinal tissue. It suppresses transcytosis, which would allow the passage of large molecules across endothelial cells into the tissue, which is damaging to the retina. And it also promotes the polarized expression of key transport systems that define barrier selectivity. And together, these coordinated effects establish and maintain the core structural and functional integrity of retinal vascular barriers.
Okay. I mean, to date, you've generated preclinical data in established animal models that show that NVQ401 can reduce neovascularization and avascular areas, and also reverse disease phenotypes that are relevant to both DME and wet AMD. Which of the sort of that in vitro data or preclinical models gave you the confidence that this is more than incremental compared to, say, for example, the current anti-VEGF therapies or other Wnt agonists that are currently in development?
So Wnt signaling targets vascular dysfunction at its root by restoring the endothelial barrier program itself, rather than attempting to counteract downstream consequences of its failure, as is the case with anti-VEGF agents, as Dr. Wong explained before. We believe that this upstream mode of action will be game-changing, not only by potentially reducing treatment frequency and duration, but also by offering meaningful benefit to current non-responders. Importantly, Wnt activation is equally effective at counteracting VEGF-induced permeability, while additionally strengthening the broader barrier phenotype. And therefore, it outperforms anti-VEGF strategies in several key aspects. In addition, as we mentioned before, our drugs are exquisitely potent, and this offers the promise of less frequent dosing than other therapies in development.
Yeah, but given the widespread involvement of Wnt signaling in multiple cell types and tissues, a major question sort of springs to my mind, and that's around safety. So what have you learned so far about the selectivity of your antibodies for the retinal endothelium? I mean, how are you de-risking sort of potential off-target effects and chronic Wnt activation?
Thank you for that question, which is really a fundamental one. So the question of selectivity is precisely what makes Neuvasq unique, because rather than broadly activating Wnt signaling or targeting frizzled receptors that are widely distributed across tissues, we modulate the pathway through a receptor complex, so the GPR124 containing receptor complex, that is selectively enriched in barrier endothelial cells. And this strategy enables precise engagement of the barrier program while minimizing off-target effects, and thereby providing a fundamentally more targeted approach to Wnt pathway modulation.
All right, so that's going to be reassuring to clinicians and also probably more importantly, patients. Why? When you first encountered the idea of targeting barrier biology more directly, through a Wnt agonist, what made this approach clinically interesting to you? I mean, what potential advantages did you see, compared to the other approaches? And actually, what type of patients would you see this approach as most useful?
Thanks for that question, Mike. Because the treatment of these important diseases, wet AMD and DME, have really relied on just a neutralization of a single factor, VEGF. And this has been the case for almost 20 years in the treatment of these patients. The discovery of a separate and different factor, Wnt, that can also on its own improve barrier function in a meaningful way was really quite groundbreaking for the field, as Ralph mentioned. And what this meant for us clinicians is that we had potentially not just one, but multiple ways of controlling the disease. And these ways can be combined for greater treatment effect. And also very exciting that Ralph had mentioned was the evidence for animal studies that retinal blood vessels which has been lost in disease, can indeed potentially be restored through Wnt action, something we know cannot be done with current VEGF treatments. Current VEGF treatments only control leakage, but does not enable regeneration and growth. So this holds the promise that Wnt agonism can help patients that are either not responsive or not fully responsive to VEGF treatments. They have recurrent swelling that comes back after many treatments, and also, importantly, have extensive damage and loss of blood vessels that would be really important and helpful to repair. So these are the factors that really increase our enthusiasm for the recognition of the second mechanism.
Right. And Ralph, while your lead program is the bispecific antibody, you're already looking at the next generation approaches and are currently developing a trispecific antibody, a molecule that combines potent GPR124 targeting with VEGF neutralization, and essentially, I guess, combining your technology with an already established standard of care therapy. So could you provide us insights into how this approach could benefit patients, as well as the strategic advantage, I guess, of this combination compared with others that are in development?
Yes. So combining these two mechanisms in one molecule, that is Wnt activation, together with standard of care anti-VEGF therapy, is expected to deliver clear patient benefit by simultaneously addressing both sides of vascular pathology. So both suppressing acute VEGF-driven permeability while actively restoring and stabilizing the endothelial barrier program by the Wnt activating portion. And this dual mechanism has the potential to enhance efficacy in difficult to treat cases, to prolong therapeutic durability, and to reduce relapse by promoting true barrier repair rather than only transient leakage control. From a translational perspective, leveraging a clinically validated anti-VEGF component alongside a complementary mechanism-driven Wnt agonist can also streamline development by anchoring safety and efficacy expectations to an established standard of care, thereby reducing uncertainty across both clinical and regulatory phases.
So why? I mean, I'd be really interested to understand or knowing how you evaluate the potential of combining these two mechanisms of action, through the tri-specific antibody. And again, which patient subgroups do you think are probably best suited for this particular strategy?
Yeah, Mike, I think the combination of both VEGF blockade with Wnt agonism can be really very significant for patients. Through the last 20 years, we know from clinical experience the benefit as well as the limitations that VEGF blockade alone can provide. And so bringing these two mechanisms together, introducing a new Wnt mechanism while keeping VEGF mechanisms on board can be potentially better than any anti-VEGF treatments we have now, and very unlikely to be worse and inferior. So this is something that is very exciting, and I'll be very interested to see how this combined treatment can just be able to bring a better response to most patients across the board, to see how we can enable a better response rate across all patients, see how that improves visual acuity even beyond what we can get from current treatments, and also, importantly, as Ralph mentioned, a more durable effect than existing treatments. I think the number of subsets of patients that are currently underserved by available standard of care treatments, these are patients that have a high level of disease activity despite frequent anti-VEGF treatments, and also patients that have lost a lot of blood vessels in the central part of the retina, which is currently limiting the visual acuity and sensitivity. So it's very interesting, at least to me, to see how adding Wnt agonism on top of current standard VEGF inhibition can transcend treatments that we have now and bring benefit, especially to these patient subsets.
So, Ralph, let's turn to the development path in terms of development timelines. I understand you're roughly 12 months from filing your first IND for the bispecific antibody, as well as close to actually nominating a clinical candidate for the trispecific molecule. And I guess 15 months from filing your first IND there. On the financing side, you're also looking to raise about 25 million euros to complete the IND enabling work, address manufacturing, and those early clinical trials with a target to, I guess, closing the financing round around September of this year, 2026. How are you positioning Neuvasq's barrier repair story to international investors, in terms of opportunity and, I guess, level of risk for the investor?
Okay, so first, there is really growing interest in the industry regarding the role of the blood-brain and blood-retina barriers in diseases of the retina as well as neurological diseases. So there is great interest in the industry and appetite, let's say, for Wnt-activating molecules. Indeed, at Neuvasq, we see a future where repair of neurovascular barriers will become a core pillar of therapy. Second, Neuvasq has, in our opinion, a very promising pipeline with different levels of risks. So there is a very solid market with a lot of interest from pharma in diabetic macular edema and wet AMD. And our Wnt activator NVQ401 represents a relatively low risk asset, I would say. Great opportunity with relatively low risk. The risk is even lower with the trispecific NVQ501, which includes the anti-VEGF functionality. And our technology can build the trend towards the use of multifunctional antibodies. And finally, there are very high reward opportunities with admittedly higher risk in neurology, given the large number of indications that could be addressed.
Great. Look, Ralph, Wai, thanks very much for joining me this episode. I mean, that was a very rich tour of a complex space.
Thank you, Mike.
Thank you.
We've heard how insights from development biology, particularly Wnt-driven, GPR124 ROT-dependent beta-catenin signaling, are being repurposed into engineered ligands and Wnt surrogate antibodies that are designed to specifically restore the blood-retina barrier function. And we've also heard how Neuvasq's lead program, NVQ401, aims to translate that into meaningful benefits for patients with vascular neuropathies such as DME and wet AMD, and also about the trispecific antibody as a potential next standard of care for those diseases. So all fascinating, and I'll definitely keep a close eye on the progress that you're going to be making. To our listeners, if you found this discussion helpful and want more conversations at the interface of deep biology and biotech business, subscribe to and share BioBiz Buzz, and also send us feedback. We would love to hear it. Until next time, I'm Mike Ward. Thanks for listening and keep on innovating.